Hi everyone, this is a continuation video of yesterday's video on the different types of bonds that form during the polymerization process of different biological molecules. And yesterday we covered it for glucose, and so we were talking about glycosidic bonds. But today we're going to look into different types of bonds in proteins and also nucleic acids as they undergo the process of polymerization. So let's get right into it. First, we're going to start off with proteins. Proteins are composed of the monomer amino acid. And essentially, when it kind of links together and forms a covalent bond, we call that covalent bond a peptide bond. So essentially, if we were to kind of link two amino acids together, that would create essentially a dipeptide molecule and also produce water, because this is a condensation reaction. So in order to explain and visually show you how this actually looks like, I'm just going to draw an amino acid molecule and also link it with another one to see the process of condensation. Right, so we have one of our amino acid molecule right here. Let's draw another one. Right, so we have two amino acid molecules. And just to kind of go as a recap, let's try to identify each of these different groups in an amino acid molecule. So this is obviously going to be an amine group. Next, we have our side chain, also known as the R group, or the variable group. Here we have our carboxyl group. And same goes for the other amino acid. So what happens is, because this is a condensation reaction, we're essentially going to get rid of the OH group right here and the H right here. And so what happens is, we're essentially going to get a structure that looks like this. Right, so we call this structure a dipeptide, and from this we're obviously going to get water out. Right? Okay, and we have essentially formed what we call a peptide bond right here. Okay, now that we have this down, let's move on to nucleic acids. Now, as nucleotides kind of bond together, what happens is the phosphate group of one nucleotide essentially bonds and forms a phosphodiester bond with another pentose sugar group of the nucleotide. So in order to kind of explain what this looks like, let's kind of recap what we learned about how the structure of a single nucleotide actually looks like. So we're obviously going to have the phosphate group. I'll just draw a quick simplified diagram. And we're going to have the pentose sugar. And then we have a nitrogenous base. Now, what would happen when several of these nucleotides bond together to form one strand? Well, what happens is we essentially get a structure that looks kind of like this. So we call each of these bonds an ester bond. And one right here as well. These are ester bonds. And because we're linking the phosphate group to the pentose sugar, we're going to call this sort of bonding in and of itself as a phosphodiester bond. Two ester bonds with a phosphate group. And this is how it's going to look like when we link several nucleotides together in order to form one single strand of a nucleotide.